Welcome to this tutorial on applied and clinical concepts related to membrane proteins, brought to you by Medical Biochemistry Power Capsule Series. This video pertains to the following CBME curriculum topic. Describe the molecular and functional organization of a cell and its subcellular components. This tutorial is preceded by Tutorial 1A and Tutorial 1B, and we recommend that you should also go through those tutorials before you watch this video. The plasma membrane and its proteins play pivotal roles in cellular function. Understanding their significance is crucial in medical science, because disruptions can lead to diseases, from drug delivery to immune response modulation. Exploring their applied and clinical importance unveils avenues for therapeutic interventions and biomedical advancements. Defects in the functioning of plasma membrane proteins can lead to a wide range of diseases and disorders, affecting various physiological processes, such as cell signaling, transport of molecules, cell adhesion, and structural integrity. In this tutorial, an overview of some of the diseases and disorders associated with defective plasma membrane proteins and their significance will be presented. In the upcoming section, you will find concise introductory remarks concerning several significant diseases within each category, given your current stage of medical education. Delving into exhaustive detail on these subjects would be unnecessary. We, however, have a mnemonic for you so that you may remember this list easily. Channelopathies are diseases caused by dysfunction of ion channels responsible for regulating the flow of ions across the cell membrane. Cystic fibrosis is perhaps one of the most well-known examples. It is caused by mutations in the cystic fibrosis transmembrane regulator gene, which encodes a chloride ion channel leading to dysfunctional chloride ion transport. This leads to abnormal ion transport across epithelial cell membranes, resulting in thick, sticky mucus accumulation in the lungs, pancreas, and other organs, and increased propensity for infections. Life-threatening cardiac arrhythmias may be caused due to mutations in genes encoding cardiac potassium ion channels and sodium ion channels. Long QT syndrome, characterized by prolonged QT interval on ECG, is one such disease. Abnormalities in neuronals, voltage-gated sodium channels, neuronals, voltage-gated potassium channels, and voltage-gated calcium channel in retina can cause the disorders as mentioned here, from epilepsy to blindness and deafness. Mutations in ion channels involved in vascular smooth muscle cell function and defective sodium channels of renal epithelium can contribute to hypertension due to excessive sodium retention and other abnormal physiological processes. Mutations in sodium and potassium channels can lead to a number of disorders affecting the locomotor system as described above, characterized by paralysis, weakness, increased muscle excitability and contractility, depending on the case. Familial hemiplegic migraine is caused by a defective neuronal's calcium channel, and congenital myasthenic syndrome is caused due to defective acetylcholine receptor in skeletal muscle. In polycystic kidney disease, polycystin 1 calcium channel is defective. Shown here is a list of channelopathies which have been briefly mentioned about in the preceding section. You would of course be taught in details about many of these diseases as you continue your journey through medical science. All of these disorders are due to gene mutations. Cell adhesion disorders result from defects in membrane proteins involved in cell-cell and cell matrix interactions, leading to abnormalities in tissue development, wound healing, and immune response. Hereditary spherocytosis is a red blood cell disorder caused by mutations in genes encoding proteins involved in the spectrin-ankyrin complex, ultimately leading to hemolytic anemia. In laminopathies, mutations in lamine proteins disrupt nuclear membrane integrity and cause tissue-specific disorders, such as muscular dystrophy and progeria. Pemphigus vulgaris is an autoimmune blistering disorder characterized by the production of autoantibodies against desmoglein proteins, which are critical for maintaining cell-cell adhesion in epithelial tissues. Leukocyte adhesion deficiency is caused by defects in integrin-mediated leukocyte adhesion. Signal transduction disorders arise from defects in membrane proteins involved in cell signaling pathways, leading to dysregulated cellular responses to extracellular stimuli. Neurofibromatosis type 1, causing benign tumors, and various rasopathies, causing developmental abnormalities and increased cancer risk, are notable examples. Diabetes mellitus type 2 is perhaps the most well-known disease in this class. Insulin signaling is mediated by the insulin receptor, a membrane-bound tyrosine kinase. Defects in insulin receptor function or downstream signaling molecules can lead to insulin resistance and the development of type 2 diabetes mellitus. Transport and receptor disorders arise from defects in membrane proteins involved in cell membrane. Transport and receptor function in familial hypercholesterolemia. Defective LDL receptor function leads to impaired cholesterol uptake and elevated blood cholesterol levels. Bartter syndrome and Fanconi syndrome are two other diseases in this category. Hartnupp's disease, cystinuria, and vitamin D-resistant rickets are other prominent examples in this category. Cystinuria is caused by defective renal tubular reabsorption of amino acids, cysteine and cysteine, from either the urine or the intestine into the blood, resulting in accumulation of cysteine in the urine, which leads to the formation of cysteine stones in the kidneys. In Wilson's disease, copper toxicity occurs due to defect in copper transporting ATPase. Paroxysmal nocturnal hemoglobinuria is another rare disease in this category. Structural disorders are diseases caused by dysfunction of proteins that are associated with maintenance of structure of cell and organelles. A few examples have been mentioned here. These diseases are not very common. Immune disorders can be caused due to dysfunction of proteins that are responsible for immunological functions in a form of severe combined immunodeficiency. The notable example in this category: mutations in genes encoding components of the immune system lead to severe impairment of T cell and or B cell function. Chronic granulomatous disease and Wiskott-Aldrich syndrome are other examples in this category. Various cancers exhibit altered expression or function of plasma membrane proteins involved in cell signaling, adhesion, and transport, contributing to tumor progression, metastasis, and therapeutic resistance. Cancers are amongst the commonest disorders caused due to defective plasma membrane proteins. We just had a glimpse at the diverse range of diseases and disorders associated with defective functioning of plasma membrane proteins. Each condition involves specific molecular mechanisms that disrupt cellular homeostasis and contribute to pathological states, underscoring the importance of understanding membrane protein biology for both basic research and clinical practice. In subsequent stages and in later chapters, you shall have the opportunity to study about the more significant ones in greater detail. In the following section, we will explore several noteworthy aspects of practical significance, encompassing few key points, from ABC proteins to poisons and inhibitors. ATP-binding cassette transporters, or ABC transporters, constitute a large family of pumps that pump amino acids, peptides, proteins, ions, lipids, biosalts and other hydrophobic compounds, including drugs, out of the cell. Defects in ABC transporters can be an etiological factor in retinal degeneration, anemia, liver failure and drug resistance of some cancers. In this animation, you can see that antidiuretic hormone is mobilizing aquaporin from their stores in epithelial cell vesicle, under influence of ADH. Aquaporins fuse with apical plasma membrane and water permeability greatly increases and more water is absorbed from the collecting duct. Aquaporin is a water channel protein. When antidiuretic hormone level drops, aquaporin 2 is again resequestered within vesicles, thereby reducing water retention. Diabetes insipidus is a rare genetic defect of aquaporin that leads to impaired water Reabsorption by the kidney, diabetes insipidus, can also be caused due to defective ADH production. Decrease in membrane fluidity may adversely affect activities of receptors and ion channels. Fluidity has a role in many diseases, such as cirrhosis, Alzheimer's disease, sparse cell anemia, alkali deficiency, hypertension, and insulin resistance. Ionophores are small hydrophobic molecules that dissolve in lipid bilayers and increase their permeability to specific inorganic ions. Most are synthesized by microorganisms. Since ionophores are not coupled to energy sources, they permit the net movement of ions only down their electrochemical gradients. They are often used in cell-related experiments and as antibiotics. Here's the list of prominent ionophores. Valinomycin is a mobile ion carrier that transports potassium down its electrochemical gradient by picking up potassium on one side of the membrane, diffusing across the bilayer, and releasing potassium on the other side. FCCP is a mobile ion carrier that makes membranes selectively leaky to hydrogen ions, and can dissipate the hydrogen ion electrochemical gradient across the mitochondrial inner membrane, thereby blocking mitochondrial ATP production. A23187 is a mobile ion carrier that transports divalent cations, such as calcium and magnesium ions. Gramicidina is a channel-forming ionophore which selectively allows monovalent cations like hydrogen, potassium and sodium to flow down their electrochemical gradients. Nystatin, amphotericin B, nitomycin are channel-forming ionophores that are used as antifungal agents. 2,4-dinitrophenyl uncouples oxidative phosphorylation from ETC by dissipating hydrogen ion gradient of mitochondrial inner membrane, thereby stopping ATP synthesis. Lipid rafts are assemblies of proteins and lipids that float freely within the bilayer of plasma membrane. They are dynamic and can cluster to form large ordered platforms, which are composed of sphingolipids and cholesterol. Cholesterol is essential for maintaining the structure of these rafts. Lipid rafts play a central role in many cellular processes, including membrane sorting and trafficking, cell polarization, signal transduction process. Research has revealed that lipid rafts have a role to play in various diseases. You may choose to go through this list as shown here. This long list is indicative of the importance of these innocuous structures found in the plasma membrane. Caveolae are cup-shaped pits in the plasma membrane of some cell types, such as skeletal muscle cells, adipocytes, endothelia, and fibroblasts. They are formed of caveolin class of proteins. Lipid rafts are concentrated in caveolae, and hence caveolae can be considered as special lipid rafts containing caveolin. Important receptors, such as insulin receptor, EGF receptor, are present in caveolae. Caveolae are associated with caveolae-mediated endocytosis, exocytosis, cholesterol homeostasis, mechanoprotective function, and signal transduction. This is a list of poisons. All of them work by blocking channels or receptors, such as sodium channels, potassium channels, and acetylcholine receptor. Most of them are extremely fatal. Wobain is a specific and potent inhibitor of sodium-potassium ATPase. Palotoxin produced by corals converts sodium-potassium ATPase into a non-specific ion channel, thereby deflating the ion gradient. Digitalis prevents efflux of sodium, thereby raising intracellular sodium. This activates a sodium-calcium antiporter in cardiac muscle. The increased influx of calcium through this antiporter elevates cytosolic calcium, which strengthens heart contractions.
Our next tutorial in the Medical Biochemistry Power Capsule series is titled Enzymology, Fundamental Concepts. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And thanks for watching this video.